Hello, today's video is going to be about carbohydrates. We are going to be explaining in detail the types, the structure, the function, and the importance they play in our bodies and in our diet. So let's kick start with the simplest of the carbohydrates known as sugars. These simple sugars are also known as monosaccharides, a word that is derived from the two Greek words monos, which means single, and the word saccharide, which means sugar. Monosaccharides generally have molecular formulas that are some multiple of the unit CH2O. For example, glucose, it has a molecular formula of C6H12O6, and it is the most common monosaccharide. It is also of central importance in the chemistry of life. In the structure of glucose, we can see the trademarks of a monosaccharide. The molecule has a carbonyl group and multiple hydroxyl groups. Depending on the location of the carbonyl group, a monosaccharide is either an aldose, an aldehyde sugar, or a ketose, a ketone sugar. Glucose, for example, is an aldose. Fructose, an isomer of glucose, is a ketose. And also, most names of sugars end in the suffix "ose." Another criteria for classifying monosaccharides is the size of the carbon skeleton, which ranges from 3 to 7 carbons long. Glucose, fructose, and other sugars that have 6 carbons are called hexoses. Trioses have 3 carbon sugars, and pentoses have 5 carbon sugars, and they are also very common. Still, another source of diversity for simple sugars is in the way their parts are arranged spatially around asymmetrical carbons. An asymmetrical carbon is a carbon attached to 4 different atoms or group of atoms. Glucose and galactose, for example, differ only in the placement of parts around one asymmetric carbon. What seems like a small difference is significant enough to give the two sugars distinctive shapes and binding activities, thus different behaviors. Although it is convenient to draw glucose with a linear carbon skeleton, this representation is not completely accurate. In aqueous solutions, glucose molecules, as well as most other 5 and 6 carbon sugars, form rings because they are the most stable form of these sugars under physiological conditions. Monosaccharides, particularly glucose, are major nutrients for cells. In the process known as cellular respiration, cells extract energy from glucose molecules by breaking them down in a series of reactions. Not only are monosaccharides a major fuel for cellular work, but their carbon skeletons also serve as raw material for the synthesis of other types of small organic molecules, such as amino acids and fatty acids. Monosaccharides that are not immediately used in these ways are generally incorporated as monomers into disaccharides or polysaccharides, which are we going to discuss next. A disaccharide consists of two monosaccharides joined by a glycosidic linkage, a covalent bond formed between two monosaccharides by a dehydration reaction. Glyco refers to carbohydrate. For example, maltose is a disaccharide formed by the linking of two molecules of glucose, also known as malt sugar. Maltose is an ingredient used in brewing beer. The most prevalent disaccharide is sucrose or table sugar. Its two monomers are glucose and fructose. Plants generally transport carbohydrates from leaves to roots and other non photosynthetic organs in the form of sucrose. Lactose, the sugar present in milk, is another disaccharide. In this case, a glucose molecule joined to a galactose molecule. Disaccharides must be broken down into monosaccharides to be used for energy by organisms. Lactose intolerance is a common condition in humans who lack lactase, the enzyme that breaks down lactose. The sugar is instead broken down by intestinal bacteria, causing formation of gas and subsequent cramping. The problem may be avoided by taking the enzyme lactase when eating or drinking dairy products or consuming dairy products that have already been treated with lactase to break down the lactose. So next up we have polysaccharides. Polysaccharides are macromolecules, polymers with a few hundred to a few thousand monosaccharides joined by a glycosidic linkage. Some monosaccharides serve as storage materials, hydrolyzed as needed to provide monosaccharides for cells. Other polysaccharides serve as building material for structures that protect the cell or the whole organism. The architecture and function of a polysaccharide are determined by its monosaccharides and the positions of its glycosidic linkages. So, storage polysaccharides, both plant and animals store sugars for later use in the form of storage polysaccharides. Plants store starch, a polymer of glucose monomers, as granules within cellular structures known as plastids. Synthesizing starch enables the plant to stockpile surplus glucose. Because glucose is a major cellular fuel, starch represents the stored energy. The sugar can later be withdrawn by the plant from this carbohydrate bank by hydrolysis, which breaks the bonds between the glucose monomers. Most animals, including humans, also have enzymes that can hydrolyze plant starch, making glucose available as a nutrient for cells. Potato tubers and grains, the fruits of wheat, maize, rice, and other grasses are the major sources of starch in the human diet. Most of the glucose monomers in starch are joined by 1 to 4 linkages, is that number 1 carbon to number 4 carbon, like the glucose units in maltose. The simplest form of starch, amylose, is unbranched. Amylopectin, a more complex starch, is a branched polymer with 1 to 6 linkages at the branch points. Most of these starches are shown in figure. Animals store a polysaccharide called glycogen, a polymer of glucose that is like amylopectin but more extensively branched. Vertebrates store glycogen mainly in liver and muscle cells. Breakdown of glycogen in these cells releases glucose when the demand for energy increases. The extensively branched structure of glycogen fits its function, is that more free ends are available for breakdown. This stored fuel cannot sustain an animal for long. In humans, for example, glycogen stores are depleted in about a day unless they are replenished by eating, and this is an issue of concern in low carbohydrate diets, which can result in weakness and fatigue. Now let's talk about structural polysaccharides. Organisms build strong materials from structural polysaccharides. For example, the polysaccharide called cellulose is a major component of the tough walls that enclose plant cells. Globally, plants produce almost 100 billion tons of cellulose per year, and it is the most abundant organic compound on earth. Like starch, cellulose is a polymer of glucose with 1 to 4 glycosidic linkages, but the linkages in these two polymers differ. The difference is based on the fact that there are actually two slightly different ring structures for glucose. When glucose forms a ring, the hydroxyl group attached to the number 1 carbon is positioned either below or above the plane of the ring. Now, these two ring forms for glucose are called alpha and beta, respectively. Also, in starch, all the glucose monomers are in the alpha configuration, the arrangement as we can see, and in contrast, the glucose monomers of cellulose are all in the beta configuration, making every glucose monomer upside down with respect to its neighbor. The differing glycosidic linkages in starch and cellulose give the two molecules distinct three-dimensional shapes. Certain starch molecules are largely helical, fitting their function of efficiently storing glucose units. Conversely, a cellulose molecule is straight. Cellulose is never branched, and some hydroxyl groups on its glucose monomers are free to hydrogen bond with the hydroxyls of other cellulose molecules lying parallel to it. In plant cell walls, parallel cellulose molecules held together in this way are grouped into units called microfibrils, as you can see them. These cable-like microfibrils are a strong building material for plants and an important substance for humans because cellulose is the major constituent of paper and it is the only component of cotton. The unbranched structure of cellulose thus fits its function, imparting strength to part of the plant. Enzymes that digest starch by hydrolyzing its alpha linkages are unable to hydrolyze the beta linkages of cellulose due to different shapes of these two molecules. Another important structural polysaccharide is chitin, the carbohydrate used by arthropods to build their exoskeletons, which are hard cases that surround the soft parts of a plant. And it is made up of chitin embedded in a layer of protein. The case is leathery and flexible at first, but becomes hardened when the proteins are chemically linked to each other. Chitin is also found in fungi, which use this polysaccharide rather than cellulose as the building material for their cell walls. Chitin is similar to cellulose with beta linkages, except that the glucose monomer of chitin has a nitrogen containing attachment. It's time you answer the next question.